It's nice to see everybody with their bright and shining faces on day 2. And what a fun day 1 was. I went home so energized, and I'm so happy to have had so many great conversations yesterday. So we have another exciting day today, and we're just gonna get right started with a keynote from Dr. Faul and Dr. Mitchell. Jessica Faul received a bachelor's in Japanese language and literature from the University of Michigan before returning to earn her master's of public health and PhD in epidemiology from the UM School of Public Health. She is a research associate professor in the Survey Research Center and the associate director of the Health and Retirement Study. Dr. Faul's work focuses on life course social and biological predictors of health and aging. Jessica also has a very long history here at ISR. She actually started at SRC in 2002 as a staff research associate hired to be an analyst of HRS's first Medicare linkage. Now to Dr. Colter Mitchell. Colter Mitchell received a master's in statistics and a PhD in sociology at the University of Michigan, and then completed a postdoc in biodemography at Princeton University. He directs the Population Neurodevelopment and Genetics program and is a research associate professor in the Survey Research Center and the Population Studies Center, and adjunct in the Department of Sociology at the University of Michigan. He leads biological data collections in the Future of Families and Child Well-Being Study, the study of adolescent to adult neurodevelopment, the M-Twin study, and the Person-to-Person Health Interview study. His work examines the influence of early life context on biological predictors across the life course. And to kind of highlight the influence of his work, Dr. Mitchell was awarded the 2017 Presidential Early Career Award for Scientists and Engineers for his work funded by NIMHD. Together, Drs. Faul and Mitchell direct the ISR Biospecimen Lab, which works with studies to develop rigorous protocols for biological sample collection, storage, and analysis. They co-lead the NIA-sponsored Biomarker Network with Dr. Eileen Crimmins, which aims to develop an interdisciplinary group of scientists dedicated to improve measurement of biological risk for late-life health outcomes in large population representative samples. And along with Dr. Aaron Ware, they direct the Genomics for Social Scientists training program, which introduces genomics and epigenetic research and analytic methods to social and behavioral scientists. So it's my pleasure to welcome Drs. Faul and Mitchell.
Nothing there yet. I'm going to let Frank handle this, and I'll start us. Well, thank you, Grace, and thanks to all the organizers. And don't worry, anyone who came here for Jessica, she will be coming up. We're going to go back and forth. I do appreciate having these sorts of activities because it does help us kind of be reminded of our shared vision, our shared experiences, history. It'll get there eventually. It's a great picture. It's really worth the wait. Perfect. There we go. See, I told you it was worth the wait. Okay, so what we hope to cover in the next 40 minutes or so could easily fill an entire book. We're talking about the life course research. And in fact, it actually already has filled an entire book. So I think Bob mentioned this yesterday. Telescope on Society was written in celebration of ISR's 50th anniversary. And if anything in our talk today piques your interest, I really encourage you to read the book, which thoroughly describes ISR's monumental impact on research in the first half century. So just as a quick outline, today we're going to briefly introduce the life course approach and share some examples of studies conducted here at ISR that have revolutionized. By the way, people in the back, someone in the back, can you hear me okay? Great. Wow, all of you did. That was great. That was a great survey. We will highlight how interconnected these projects are. That's going to be one of our key themes with the work being done here at ISR, essentially tracing the life course or evolution of these studies. Furthermore, we'll discuss kind of some critical methodological innovations that have driven the field forward and end with some exciting work currently underway. However, regrettably, we can't delve into very much detail on any one study. And even more importantly, due to time constraints, we won't be able to cover some projects and many people we won't be able to mention. So we sincerely apologize for that, but luckily there are some sessions immediately afterward. So we encourage you to participate and celebrate those other projects. Finally, we have two major takeaways that we want you to remember. One is that ISR has been central and continues to be central in the growth of life course research. Second, all of the studies, all of the methodologies we discussed today are a result of a collaborative effort across various fields and areas of expertise, and I just don't mean academic disciplines. At ISR, we build off of each other's work, driving innovation and excellence together. I have to smile a little bit. I'm going to talk about the life course approach, but I studied life course approach, and we had discussions about it in the PSC library, which was right over there, before this atrium even existed. So it's just kind of funny to be able to talk about it so close to where we studied it. So the life course approach, also known as life course theory or life course perspective, has evolved into a powerful framework for analyzing people's lives within structural, social, and cultural contexts. Although its roots actually go back to someone like W.I. Thomas in the 1920s, it gained significant rigor and traction in the 1960s and 70s, and that's a key time. This approach views individuals' lives as socially sequenced timeline, emphasizing historical events and social changes. So, for example, Glenn Elder's example of the Great Depression, or more recently, COVID-19. And if you think about how that influenced, say, kids who were just starting school versus high school students versus someone looking for a job versus someone about ready to retire, pretty different experiences for people. It's also been instrumental in elucidating health inequalities, demonstrating how early life conditions can profoundly influence adult health outcomes. The life course approach has garnered interdisciplinary attention, seamlessly integrating insights from psychology, sociology, history, economics, and public health. Most critically for today's discussion, the Institute for Social Research has been at the forefront of this field. As you could see, there was a desperate need in the 1960s, especially in the 70s, for test longitudinal data that was needed to test these life course theories and hypotheses, and ISR provided some of the first and certainly most widely used longitudinal data. It's also been a pioneer in creating and refining novel methods for measuring experiences and exposures across the lifespan. And its commitment to sharing the data with the broader research community has catalyzed countless studies and advancements in life course research. So it filled that gap. Okay, so now we're going to turn, I told you it was going to be brief, we're going to turn to some of the examples, some of the studies. And to go back, let's go back to the very first decade of ISR's existence and talk about two key projects that eventually lead to ISR's first birth cohort study. So in 1951, Ron Friedman, founder of the Population Studies Center, and Angus Campbell, who was the first director of SRC, you heard a little bit about them yesterday, initiated the Detroit Area Study, which ran repeated cross-sectional surveys annually from 1952 until 2004. The DAS was representative of the Detroit metro area, and it was designed for three things: to train students in survey methodology, provide vital scientific information about the Tri-County area, and generate novel data collection opportunities. I am really quite proud to have been a student trained in DAS. However, I'm a little sad that I was in the last cohort. Just out of curiosity, how many here were trained in DAS or worked on a DAS project at some point? Okay, good. So, today, by the way, the Detroit Area Project, the influence of that project can be seen in the, by the way, this is the book if you want to get a book about it too. They're hard to come by though, I found. The influence can be seen in the Detroit metro area community studies, or DMACS, which provides timely survey data to Detroit and is run by ISR faculty member Jeff Mornoff. So here's the other study, and I'll move quickly through this one. This was the SRC's Growth of American Families, or GAF, which was conducted in 1955. This was the first national sample survey of fertility. I know that sounds crazy, but that was the first one. And the impact was so significant that they repeated it in 1960. And this leads to the National Fertility Study and ultimately the National Survey of Family Growth, which was another big ISR product. But one of the interesting things that happens is Ron Friedman recognizes that in order to test the life course approach, to think about these things, you can't just have cross-sectional studies. We need a panel study. We need a birth cohort study. So Ron Friedman, David Goldberg launched the Family Growth in Detroit study. This study interviewed women in the Detroit area who had either recently been married or given birth to their first, second, or fourth child in July of 1961. And what did they use to collect it? The Detroit area study in 1962. So January through March of 1962, follow-up interviews in the fall of 1962, 63, and 66 make it one of the very first successful birth cohorts in the United States. Then Arlen Thornton and Deborah Friedman take over for the study in 1977. They interview the mothers again, and then they change things again. And they turned it into the intergenerational panel study. And how is it called that? Or why is it called that? Because in 1980, 1985, and 1993, they interviewed the mothers and the children. The resulting longitudinal study encompasses 8 waves of data collection from mothers and their offspring through age 32. So more broadly, the intergenerational panel study showed that young adults are influenced by multiple dimensions of their parent family through mechanisms such as socialization and social control. So things like parental attitudes, values, socioeconomic status, employment, marital experience, and religious affiliation are all studied. For example, parents' values influence offspring behavior by socializing or influencing their kids' own behaviors. That makes sense. But they also have independent effects, even controlling for their parents or their offspring's values. But far beyond its own results, the intergenerational panel study influences the creation of the Future of Families and Child Wellbeing study in 1998, which is the only still running U.S. national birth cohort of around 5,000 families. And bringing it full circle, I'm very happy to say the Survey Research Center, SRO, is collecting a COVID supplement right now on the Future of Families, and we'll be collecting the year 27 data. And through the Future of Families and Child Wellbeing study, it also has influenced, the intergenerational panel study has influenced the Healthy Brain and Child Development study, which I'm guessing most of you don't know about, which is a current initiative or current project actually being run by NIDA to start a new national birth cohort of around 7,500 families, focusing on the effects of substance use on birth, infant, and child health. Okay, so that's one set. We're going to keep going. Also in the 1960s, another group of scholars turned their attention to the impact of education on adolescent and young adult trajectories, particularly focusing on disentangling selection and socialization. And during this area, several studies emerged to study the role of high school education, including the Youth in Transition project, or study, led by Jerry Bachman. All of these education studies are pretty unique, and they teach us some key things. Number one, not surprisingly, we need longitudinal data to do this. Number two, it should be nationally representative so that the results are generalizable. Number three, we actually need a sufficiently large study to be able to study relatively rare events. It's really important to consider the context of the data collection. And we need a multiple cohort design in order to discern the differences between cohorts and age. Makes sense. From this emerges the Monitoring the Future study, led by Lloyd Johnston, Jerry Bachman, and Patrick O'Malley, which was designed to study changes in beliefs and attitudes among young Americans. So I think probably most of you have heard of MTF. It's renowned for its national estimates of legal and illegal substance use. And it began surveying seniors in 1975 and expanded in 1991 to include 8th and 10th graders from approximately 400 schools annually. Think about that coordination effort. To date, MTF has surveyed over 1.7 million students. It's led by Richard Miek. And for those who haven't yet explored the repeated cross-sectional reports, I actually highly suggest you go on their website and do that. They have invaluable number of or data and tracking numerous variables over now almost half a century. And it provides a lot of information on changing patterns in substance use and highlighting shifts in attitudes and socioeconomic influences. Okay, so this talk, though, is about life course approaches. So I want to delve a little bit deeper into the MTF panel study. Again, I'm wondering how many of you know about the panel study. So it was initially led by Lloyd Johnston, then John Schulenberg, and now Megan Patrick. The MTF panel study extends the work of the main study by following a subset of those MTF seniors. If you think about it, that final year of high school, or high school in general, is an incredible time. It's a time of major transition from adolescence to adulthood. So it's a great time to start a study like this and get some insights. So starting with the class of 1976, around 2,400 randomly selected graduating seniors have been surveyed every 2 years until age 30, and then 30 and on, it's every 5 years. The study has started with 120,000 MTF seniors, so pretty sizable cohort or study, and now they have people up to age 65. So pretty impressive. The study has shown that substance use persistence patterns established during adolescence often persist into adulthood, with early initiation linked to ongoing use and potential substance-related problems. It has also demonstrated that major life transitions, such as entering college, starting a career, or getting married, strongly influence substance use patterns. But beyond its direct findings, which again, I warned you all, we can't talk about all these studies in detail. MTF has significantly influenced other studies through its reports, publications, but in addition to that, it has had influences on other studies, like here at CPS, we have the longitudinal study, we've had the longitudinal study of American youth, and more recently, the Adolescent to Brain and Cognitive Development Study, or ABCD, I think we heard a little bit about that yesterday. This is a study, a current study of around 12,000, almost 12,000 students that started when they were age 9 and 10, and MTF's estimates of help determine the optimal starting point for examining how drug use impacts brain development, underscoring the large and far-reaching impact of monitoring the future projects.
Hi, everyone. So I'm going to continue with a little bit of our history here. In the 1960s, at the same time the intergenerational panel study was getting established and the seeds of what would become MTF were sown, interest was growing in behavioral economics, focusing on inequality, family income, and wealth, and how circumstances were passed down across generations. The Census Bureau was tasked with assessing the effects of President Johnson's war on poverty. This led to the Survey of Economic Opportunity, which interviewed 30,000 households, first in 1966 and then again in 1967. The Office of Economic Opportunity then sought a university to continue this work. They wanted to follow academic trajectories of low-income families. While OEO wanted to focus solely on low-income families, Jim Morgan, the founding director of the PSID, or Panel Study of Income Dynamics, argued for a population-wide scope. He emphasized the importance of understanding not just how families fall into poverty, but also how they rise out of it. Okay. The PSID began in 1968 as a nationally representative annual survey of families, and since 1998, it has been conducted every 2 years. In 1997 and again in 2017, it added immigrant refresher samples to include families who had immigrated to the United States since 1968. Its genealogy-based design invites children and grandchildren of the sample families to join the study when they form their own households. Deciding on this innovative design was a huge event in survey design history. This approach provided continuous, self-representing samples and allowed the study of intergenerational connections. So we're not just following a life course, but the life course of entire family lines. Over time, the PSID evolved from a study of poverty to a study that was a rich longitudinal resource for social scientists, covering employment, income, wealth, health, education, marriage, childbearing, and more. Today, with over 55 years of data on those individuals and families from the 1968 sample and their descendants, the PSID remains the longest-running longitudinal household survey in the world, offering unparalleled insights into intergenerational dynamics and social mobility. The PSID explores more of the life course through its supplements. The Child Development Supplement provides data on the PSID children and their extended families in order to study early human and social capital formation. The Transition into Adulthood Supplement, or TAS, began in 2005 to follow children from the original CDS cohort into young adulthood, and now follows all PSID sample children who are entering early adulthood. They have additional supplements on aging, disability, intergenerational transfers, and well-being, and these have been collected to give a more complete picture of the events and influences that shape our lives. Intergenerational dynamics learned from PSID are crucial to life course theory. They help us understand how parents' economic status and decisions shape their children's opportunities and outcomes over time. One key finding from the PSID is that social and economic trajectories are far more varied and unpredictable than initially thought. While average trajectories across age groups look predictable, longitudinal data show that individual paths can diverge quite a bit from these averages. Also, early PSID measures, such as personal control and achievement motivation, have proven to be long-term predictors of intergenerational success. And by tracking significant life changes like employment, marriage, childbirth, divorce, and retirement, the PSID offers valuable insights into how these transitions shape individual trajectories. And the PSID also supports the life course theory concepts of cumulative disadvantage or advantage, showing how early life conditions, such as parents' education, impact long-term economic outcomes. The success of the PSID has inspired similar studies worldwide, including the British Household Panel Survey and Swedish Household Panel, to name a few. And their innovation continues with their recent expansion into genetic data, allowing us to investigate the effects of nature versus nurture by collecting genetic data from families, specifically siblings. This ongoing evolution ensures that PSID remains a vital resource for understanding life course and intergenerational dynamics. But we didn't just do that. We have more investigation. In the 1980s, life course research saw an expansion again. This time, the lens expanded from a focus on the family to social networks, including friends and family more generally. Bob Kahn and Tony Antonucci started the Social Networks in Adult Life study, a national probability sample of men and women ranging in age from 50 to 95. And they also presented their lifespan development model, the convoys of social support. And this was a way to describe interpersonal relationships over time and their effects on health, well-being, and quality of life. They describe 3 concentric circles of relationship closeness, which vary in their stability over the lifespan. And they describe the different types of support that could be exchanged through these relationships. The study of social context expanded again with the start of Americans Changing Lives. The study began in 1986 with a national face-to-face survey of over 3,000 adults. It is the longest ongoing national study that examines how our neighborhoods, work, and social connections affect our health throughout adulthood. and more, the Survey of Black Americans. Since the initial National Survey of Black Americans in 1979, the Program for Research on Black Americans has conducted numerous quantitative and qualitative studies to understand the lives and experiences of Black Americans. This landmark study used a sampling technique that, for the first time, made African American households, even those living in areas with small populations of Blacks, eligible to participate in the national survey, thus providing the first truly representative look at Black Americans. And now we turn to our final example, the health and retirement study, which arose from a pressing need for comprehensive data on aging. The Retirement History Survey was a project of the Social Security Administration. It was conducted by the Census Bureau starting in 1969. It tracked around 12,000 individuals, but was limited to men and unmarried women born between 1906 and 1911, with a short follow-up of only about 10 years. As women increasingly joined the workforce, it became clear that more diverse and extensive data were needed to understand work and retirement comprehensively. Similarly, the study of aging as a process was evolving. Early aging research like the Seattle studies and the Baltimore longitudinal study of aging revealed significant variability in aging, showing that things like health and functioning did not always decline with age. There was quite a lot of heterogeneity there. And so emerged the demography of aging, the idea that in order to understand this part of the life course, we were going to need to integrate demography, economics, epidemiology, psychology, and sociology. And so created by an act of Congress in 1990, the HRS became the first longitudinal study to incorporate detailed economic and health information on older adults in the same survey. This groundbreaking initiative has provided invaluable data, transforming our understanding of aging in the United States. The HRS began data collection in 1992, led by Tom Juster. The collection occurs every 2 years on approximately 20,000 people, with the aim of creating a data resource from a nationally representative sample of Americans 51 years and older. The data captures the evolving health and economic circumstances linked to aging at both the individual and population levels. The HRS brought together a multidisciplinary team, addressing that this was what was needed to study the complex interrelationships between the factors that affected work, health, and family at older ages. The steady state design added a birth cohort every 6 years to maintain representativeness. And oversamples of minoritized groups were added, allowing for well-powered investigation of race and ethnic subgroups. This became a focus of the HRS. It provided a resource for methodological studies on survey non-response patterns, interviewer and mode effects, and it also led to innovations that moved aging research forward. The measurement of cognition in population-based surveys, the collection of physical measures and biomarkers, the creation of harmonized polygenic scores from DNA data, and the addition of administrative linkages and contextual data. These things were showing up in other studies too, but as David Weir, the current co-director of the survey, said, figuring out how to do those things in a large, diverse, and representative population is what makes HRS HRS. And now you.
See, we're just passing back and forth. Just don't worry, she'll come up again. Although there were many methodological life course innovations that have already been alluded to, including statistical methods, survey methods, measurement expansions. We want to talk about two that were critical here at ISR, the life history calendar and biomarkers. And Jessica just wanted me to point out something. So we have a lab that's downstairs right over here, and people often think of it as the first lab. In fact, there was another lab, and I think I heard today that it was on the 4th floor in this building. So it did exist before, that's why that's there, just so everyone knows. Okay, so let's talk a little bit about the life history calendar. Although prospective longitudinal studies collected at short, regular intervals, like we've been describing, is really great, right? That's kind of the dream scenario. Logistically and cost sometimes doesn't allow us to do that. And so there are barriers that make that impractical. A groundbreaking methodological innovation for life course research was developed here at ISR, and that's called the Life History Calendar. The Life History Calendar enhances the accuracy and reliability of retrospective data collection by providing a visual and structured way for respondents to chronologically organize and report their life experiences. So I don't know how many of you have seen a life history calendar. This is not really a fair look. They can be quite large. This visual layout, typically featuring grids and timelines, helps respondents anchor their memories around significant life events, thereby improving data quality. So if you can imagine, if I just asked you a series of questions of what date did you start this job, what date did you get married, what date did you get divorced, what date did you move to this place, that can actually be quite taxing. So the method that they use is you can visually see the starts and stops, you can report the dates that you can remember, start there, and then you can probably anchor, was it before that, was it after that, how long before? And so it's actually quite helpful, but it takes a lot of back and forth between the interviewer and the respondent. So although earlier versions of the Life History Calendar appeared somewhat in the 1960s and 70s, its refinement and significant application by Deborah Friedman, Arlen Thornton, Dwayne Allwyn, and Linda Young DeMarco in the 1985 intergenerational panel study, see, right back around. So those interviews truly set the standard. This method has been widely adopted across disciplines, including sociology, psychology, epidemiology, public health, to study things like family dynamics, educational attainment, health behaviors, and employment histories. So there is a series of notable adaptations to the Life History Calendar that can be seen in the Chitwan Valley Family Study, which was launched in 1995 with over 5,000 individuals in 171 neighborhoods in Nepal. So ISR's Bill Axon and Dirga Gamiri and others adapted the Life History Calendar for the Nepali context by incorporating some additional time measures, including the Nepali year, some national and community events, so was it before or after an earthquake, and the animal year. And more recently, it was modified again to more accurately measure mental health timing and levels in the Chitwan Valley Family Study. Also, because it's a key focus of CVFS is contextual measurement as people move into the neighborhoods as they join the study. And as of right now, they've collected over 10,000 life histories. In addition, I think one thing that's particularly unique is that CVFS has used the same life history calendar technique to measure things like the birth and growth and change of a neighborhood, or the schools, or a health post, so using the same techniques. And I think that was the first time I've seen it applied at that other level. And just to bring this back, PSID, HRS, and other studies have effectively used the Life History Calendar to get the experiences of respondents, say, prior to age 51 for HRS. What's interesting is that it continues to be adapted, the Life History Calendar, including now work on a web-based version, which kind of demonstrates the invaluable role of capturing detailed life histories and enhancing our understanding of the complex interplay of life experiences over time. Okay, so let me turn to a very different thing for a second. I mentioned the lab before, so you get a little bit more of biology for a second. Another rapidly expanding area in the life course research is the use of biological data. Often when we talk about biological data, we think of it as a mechanism, right? Linking an exposure to health, and it operates through some biological mechanism. Or potentially we think of it as explaining why there's variable responses to an exposure, to the same exposure. However, I would argue that the most successful that we've been in using biological data in our studies has been as a biomarker. So these are biomarkers which may not be on the causal path, but can identify disease before diagnosis. And sometimes we have chemical residues in our bodies that can reflect past exposures. So essentially, these biomarkers allow the researchers to potentially recreate an unmeasured past and predict the future. A particularly valuable type of biomarker in life course research that's currently being studied at several projects here at ISR is an epigenetic surrogate biomarker. So epigenetics, I promise this won't last long. Epigenetics involves chemical modifications of DNA without changing the DNA sequence that influence gene expression, and importantly, can change with time and with exposure. A prominent example of this is an epigenetic clock, which you probably have heard of. It's on Time magazine, so it's out there, which often uses large amounts of epigenetic data to predict aging, morbidity, mortality. And clocks can reveal something that people call biological aging, how much older or younger one's body is compared to what you would expect given their chronological age. So interestingly, other epigenetic surrogate biomarkers are specific to conditions or processes. For example, inflammation or cardiovascular disease or BMI. These are often developed in older adults using, again, machine learning techniques of hundreds of thousands of epigenetic measures. And the interesting thing is they then can be applied to younger populations where potentially life course, early life context is more thoroughly measured. As an example, you can train something in the health and retirement study where we have a lot more variation in health outcomes, and then recalculate those using epigenetic measures in the future of families, the birth cohort. And what we're seeing is that even in child samples, these surrogates developed in adults often predict disease in kids, even controlling for close approximations for disease. For example, BMI epigenetic surrogate predicts later BMI, even controlling for current BMI and current health. And what's really fascinating is you see very strong correlations between the surrogates, these adult trained surrogates, and early life context, which helps us link together early life and later life. Another way that we do this is that we take research being done here is also showing that there are links between early life context measured with epigenetic surrogates and later life disease. So in this case, what you're talking about is taking something in a child study, like the Future of Family study, and applying it to, say, an adult study like HRS. For example, maternal smoking during pregnancy. We can see that signature, the epigenetic signature, in the offsprings at birth and in childhood and adolescence. We can see that signature in the Future of Families. And those same scores, when applied to older adults, even accounting for their own personal smoking behavior, predict later life health. So similarly, exposures such as to lead, to pollution, to adverse childhood experiences are being studied to recreate these unmeasured past exposures in older adult populations and to examine kind of the long reach of early life exposures. And in some ways, if you think about it, there are certain childhood contexts that even if we sat down with a life history calendar, you might not be able to ever recall. So just as to kind of conclude on this, by utilizing epigenetic measures, researchers can essentially condense that survey period, better estimate the unmeasured past, predict disease and aging trajectories more accurately. And this innovation significantly enhances our understanding of the long-term impacts of early life exposures on health and development. And now that they are added to more nationally representative studies, particularly here at ISR, we're also doing one other thing, which is we're making biosocial literature more generalizable, something that it has actually desperately struggled with.
And the innovations in life course research are still ongoing today at ISR. New data are being collected, and we are creating new techniques to continue to gather those data. In addition, tremendous innovation has occurred to improve and expand the measurement of the social and environmental context. For example, we're working on linking historical records, data on pollution, neighborhoods, and policy with time and geographic specificity that revolutionized this area. And again, much of that has been done here with the development of resources like the Census RDC, NANDA, and the Michigan Center on the Demography of Aging's Geographic Linkages Repository. We are also pioneering research on the effects of climate change and structural racism using both surveys and geographically linked data. We are expanding in the inclusion of the voices we represent. This has always been a hallmark of the research at ISR, and it continues today, with ongoing studies and studies in development that focus on the experiences of Arab and Asian Americans, as well as Alaska Natives and Native Hawaiian communities. But potentially the single most important contribution ISR has made to life course research is the sharing of the data we collect here. It is not by accident that the story of life course research and its development almost exactly mirrors the development of studies here at ISR. Our data were the driver. We provide data for public use. It is what we do. And it is what ICPSR was founded to do. They were established in 1962, and they serve as a vast archive of digital social science data, providing researchers, policymakers, students, educators with access to a wide array of data sets covering numerous domains. Many impressive studies have limited or no outside access to their data and ultimately struggle to make as large an impact on the field. Our impact can be seen by looking at the number of publications that cite our data. For example, the PSID has 92 books, 1,291 dissertations, 84 reports, and over 53,000 journal articles cited. HRS has 236 book chapters, 839 dissertations, 1,209 reports, and 5,377 journal articles cite their data. And ICPSR has almost 21,000 studies, 6.5 million variables, and 23 specialized collections, resulting in over 116,000 publications. ISR's influence in life course research is unparalleled due to our extensive collection of data, specifically longitudinal data, which tracks individuals and groups over significant periods in their lives. Our data enables researchers to explore the dynamic processes and transitions that shape individual life trajectories. And the data sets allow for in-depth analyses of how these early life experiences, socioeconomic factors, health conditions, and policy interventions impact outcomes across the lifespan. At this point, we have highlighted studies over 7 decades, showcasing the breadth of ISR's impact. And in true ISR fashion, we've navigated through a myriad of acronyms. And while it's easy to marvel at these projects individually, one fact stands out. These studies were exceptional, in part because they were conducted here at ISR. Each project was built on the pioneering work and innovative ideas that came before them, creating a rich tapestry of overlapping expertise and knowledge. The real story lies in the collaboration and shared purpose amongst us all, much of which goes undocumented but is no less important. Analysts and project managers trained on groundbreaking projects now share their expertise with others, while programmers continue to push the envelope of technological advancement by working together across centers and projects. Our well-regarded grant managers train and mentor the next generation of experts, and survey methodologists apply cutting-edge techniques across projects. Our data collection staff and seasoned interviewers take on the toughest cases in the field and pass on their skills to future generations. The culture of collective effort and shared success is what makes ISR a powerhouse in life course research. Together, we push the boundaries of what is possible, learning from both our success and our challenges. And although it doesn't always feel so grandiose in any one particular moment, when observing ISR through a historical lens, it is clear this is who we are at our core. And Colter and I think that this is what will guide us into a successful future. Thank you.